This is Vrinda V and I am currently doing my BTech in Biotechnology from Vellore Institute of Technology. And here I am again to continue with the chapter, the fundamental unit of life. So, I am expecting and supposing that you have worked on the previous lesson and you have read the textbook and learnt all the topics. So, with this, let's move forward. So, as for today's agenda, we are going to discuss something about microscope, and we are also going to learn about cell organelles with specific focus on plasma membrane. Okay, so before we get started, I have a question for you again. So, last time we learned who is the father of biology, right? So, this time I want to ask you, who is the father of plant biology? You have a few seconds to make your guesses. I want all of you to think about it, make your guesses in the mind. Alright, so the father of plant biology is Theophrastus. So, Theophrastus had done a lot of study about plants and finally he wrote 2 books on plants, which is the history of plants and on the causes of plants. So, these books had so much immense knowledge that it became very difficult for scientists to find any new discovery the next 1800 years. Everything that they discovered was there in the book. Okay? Alright. And he was the one who introduced agriculture to botany. And that's why he is known as the father of botany and father of plant biology. Okay. So now, there's a lot on today's agenda as well. So let's get started. So let's start with microscope. Okay? So, microscope is a device which is used to magnify objects, that is, see smaller objects on a larger scale. So, the first ever microscope, which is very simple and it might be difficult for you to believe if I tell you what it is. Do you know what it is? It's just a simple magnifying glass. Hard for you to believe, right? So, that's where it all started, with a magnifying glass. Okay, then sooner or later, compound microscope was discovered and a picture of compound microscope is given in your textbook in the first page of this very lesson, that the first diagram, that is the figure 5.1. I want you all to refer the figure 5.1 to see a compound microscope. So there are several parts. You don't need to learn the whole mechanism of the microscope. Just know that these parts are called as these in a microscope. Alright. So there's an eyepiece, okay, and there's a coarse adjustment, fine adjustment, basically to focus, and there's an arm, there is a base, there's a mirror at the base, okay, there is a stage. So there are a lot of other parts. Just go through the figure 5.1. Alright, and then the next microscope was discovered in 1940, which changed a lot of things about cell and cell biology. That was electron microscope. So, after discovering electron microscope, people came to know that there are several parts inside a cell too. So, do you remember that last class I told you about the cells performing several functions and cells are responsible to perform so many functions? Then, who is actually performing all these functions for cell? There should be something inside the cell, right? Which is performing all these functions. So, that is nothing but certain components of cell which are called as cell organelles. So basically from now on our main study area will be all about this, that is cell organelles. Okay. So when they observed it under the electron microscope and when they saw the cell, they mainly observed 3 features. So what are the 3 basic features that every cell has? I'll show it to you. So the 3 basic features that every cell has. One is the outermost covering of the cell, which is called as plasma membrane, and it's also known as cell membrane. The other is a sphere inside a sphere, that is a small sphere inside a sphere, that is a cell. It need not be a sphere, okay? So it's something inside in the center of the cell and it's called as a nucleus. And the rest of the area inside the cytoplasm inside the plasma membrane, and outside the nucleus is called cytoplasm. So there are various substances in the cytoplasm, and it's a vast topic, and we will be talking about it in the next class. Alright, so for now let's focus on plasma membrane and nucleus. Talking about plasma membrane is the outermost covering of any cell. So basically, plasma membrane is a substance that decides that what should come inside the cell and what should go outside the cell. So it allows and prevents the entry of so many substances in and out of the cell. Like a sieve, you know, you use a sieve to filter out the tea. So there are certain substances that are collected on the filter, on the sieve, right? Like the tea powder which does not go down with the tea, and only the tea that you drink, the liquid, goes down. Similar to that, plasma membrane basically acts like a sieve. So it filters out so many substances. It only allows the transfer of some substances in and out of the cell. Okay, so we can say that it selectively permits the entry and exit of substances in and out of the cell. So that's why it's also called as selectively permeable membrane. Alright, so it's called as selectively permeable membrane. Okay, so now how is this transfer of substances occurring inside and outside of a cell? So that is basically through so many kind of transport mechanism, and one such mechanism is diffusion. So diffusion is the movement of substances from a higher concentration region to a lower concentration region without the use of energy. For example, if I have something here which is in higher concentration and here in low concentration, then substances will move from here to here, from higher concentration to lower concentration without the use of energy. Such kind of movement is called as diffusion. So basically all the gaseous exchanges occurs in a cell through diffusion. Basically the exchange of carbon dioxide and the exchange of oxygen occurs through diffusion. I will show you how. So in a cell, we know that oxygen is converted into carbon dioxide. So oxygen enters and it's converted into carbon dioxide, and soon so many products, so many carbon dioxides are produced, molecules are produced inside the cell, and relatively lesser carbon dioxide molecules are outside the cell. So carbon dioxide moves from its higher concentration region to its lower concentration region. The same thing occurs with oxygen. As and when carbon dioxide is produced, this oxygen gets depleted. So there is less oxygen inside the cell than outside the cell. So oxygen enters inside the cell from out of the cell. So that's how the transport of oxygen and carbon dioxide occurs in and out of the cell through diffusion. Now, how does water molecule move in and out of the cell? So water molecule has a specific mechanism for this, which is called as osmosis. So osmosis is similar to diffusion, but the only difference is it is specifically for water. So now why is there something very specific for water alone? Because you know what? Osmosis also occurs when water moves from higher water concentration to lower water concentration. But we know that everything in our body is dissolved, either dissolved or not dissolved in water, right? So supposedly there is water over here and there is water over here, alright? And so many substances are dissolved here. Relatively lesser substances are dissolved here. So can we call this a concentrated solution? And can we call this a dilute solution? Yes, right? But here there is less concentration of water and here there is more concentration of water, right? So how will osmosis take place? It will take place obviously from higher water concentration to lower water concentration, because what is moving is water. So it will move from higher water concentration to lower water concentration as per the definition of diffusion without the use of energy. But how is it actually moving from a dilute solution to a concentrated solution? You got the mechanism, and that's why there is a specific name for the transport of water, which is known as osmosis. This osmosis occurs in various ways, and depending on the environment the cell is exposed to. So I'll tell you how this occurs. So supposingly a cell is placed in a medium where the concentration of water outside the cell is more than the concentration of water inside the cell. That is, the cell is placed in a dilute solution. So water molecule will obviously move outside and inside of the cell, but the net movement of water molecules will be more from outside of the cell to inside of the cell since it's placed in a dilute solution and osmosis and diffusion occurs. Alright, so this results in swelling up of cells and this is called as endosmosis, and such type of solution is called as hypotonic solution. So endosmosis occurs and such type of solution are called as hypotonic solution. Alright, so another case is where a cell is placed in a medium where the concentration of water is very less and outside and concentration of water is more inside of the cell. That is, it's placed in a concentrated solution. So in such a case, the water molecule, the net movement of water molecule will be more from inside of cell to outside of cell. And such type of movement of water molecule is called as exosmosis, and such a solution is called as hypertonic solution, and in this case a cell shrinks. And there is a third case where the amount of water in and out, outside and outside of the cell is almost the same. So there is no net movement of water molecule from in to out or out to in. Alright, because the equal number of water molecules moves outside and inside at the same time. So such type of osmosis is called as a neutral osmosis, and such in cells in such a state are said to be in equilibrium. Alright, so we learned that there can be exosmosis, endosmosis, and there can be a neutral kind of osmosis where the cells are in equilibrium and all that. And now, do you know, hearing all that, aren't you excited to observe osmosis by yourself? And I tell you that you can actually do this. All you have to do is take an egg, put it in a dilute hydrochloric acid solution, and so what happens is the shell of an egg is made up of calcium carbonate. So when you put it in HCl solution, it dissolves. Alright, and it leaves a thin sheet covering the egg. Take a thin sheet covering the egg. You put one egg in salt solution and another egg in water solution. You'll observe that the egg that is put in water solution swells up due to endosmosis, and the one put in salt solution shrinks due to exosmosis. The similar thing can be observed with raisins as well. You can easily do this experiment at your home. Just take some raisins, put some raisins in water solution and some raisins in salt solution. You'll see the raisins put in water solution swells up and the raisins put in salt solution shrinks. So this is all about osmosis. Now there is another mode of transport that occurs in a cell called as active transport. So active transport usually occurs when in nutrient molecules. So there are certain nutrient molecules that need to move from a lower concentration to a higher concentration, and that requires energy. So the movement of substances in and out of the cell from a lower concentration to a higher concentration by utilizing energy is called as an active transport. So that basically occurs in certain nutrients. Alright. Okay. So there's a fourth mode of transport that you have already learned in the previous lesson. So that is called as endocytosis. So what happens in this endocytosis is, do you remember the case of amoeba? Where amoeba extend its pseudopodia and engulfs the food material. So this is what it is called, endocytosis. Because this happens because the plasma membrane of amoeba gets extended. This happens because the plasma membrane is flexible. So plasma membrane is made up of lipids and proteins. So this makes the plasma membrane flexible. So the movement or engulfing of food substances or the movement of substances inside a cell, which is facilitated by the flexibility of plasma membrane, is called as endocytosis. So this is all for today's lesson. So in today's lesson you have learned about microscope and a few modes of transport like diffusion, osmosis, active transport, endosmosis, and of course about plasma membrane. In the next session you will learn a lot about the different types of cell organelles, about nucleus and about cell division, and it's a lot more exciting. So for now I want you to go through this lesson, read the textbook, learn the topics, and yes, of course, do that experiment of freezing so that you can observe yourself how osmosis is actually occurring. Alright, so see you in the next class. But yeah, if you have any doubts going through the lessons, you can always post your doubts. We'll get back to you as soon as possible, possibly within 24 hours. Till then, take care.